Okay, this is my video on meiosis. Some key differences from meiosis to mitosis is that this is for gametes, so like sex cells like sperm and eggs. They produce genetically unique cells, like they are not identical to the parent cell. They produce half the number of chromosomes, so they all will be haploid cells. And it undergoes meiosis twice, so the outcome will be 4 daughter cells, unlike mitosis, which only has 2. And then there will be 4 haploid cells. They are haploid because they're only coming from either the mother or the father, and there won't be no replication of either or. There has to be the mixing of the sperm and the egg for it to be a total number of a diploid cell. And another important key part of meiosis is crossing over, because that's what makes all the cells genetically unique. It divides each cell by each chromosome by the genes, so the chromosome can look completely different as long as the genes are located like in the spots that they're supposed to be expressed. So it's kind of the same parts of meiosis. There's prophase, metaphase, anaphase, telophase, and cytokinesis. It just goes all of it twice. So prophase is when, like, we already have interphase because I talked about the cell cycle before, and we have the whole cell cycle, but instead of mitosis, we're just going through meiosis. So synthesis and replication has already occurred. So then we have prophase and the condensing of the chromosomes and the spindle fibers being created. I didn't write that, but the spindle fibers are being created to go there. And then metaphase is when the spindle fibers attach to each centromere of the chromosomes, and crossing over happens in prophase, I believe. Crossing over is happening in one of these. Yeah, crossing over happens in prophase. And then we have metaphase, which is the alignment of the crossed over chromosomes. And then anaphase, when they go to opposite sides, polar sides of the cells. And it's random now because they are not identical to the parent cells, so they are random, these sex cells. And this genetic information, although they do come, like it has the same genetic information of the parent cell, there are certain aspects of randomized. Then we have telophase, which is the nuclei is coming back, because we know that nucleus were broken up in prophase. And then we have the first, well, this is separated. So we have, this is prophase and mitosis. This is all mitosis 1, meiosis 1. And this is meiosis 2 on this side. So then after we have prophase, one of these cells will go and they'll cross over again to do prophase 1, prophase 2. I'm really tired, but they'll cross over to prophase 2, and then they're different. You see how these cells, these chromosomes look different from these ones? Because they are different. They are not genetically, like they are genetically different. They're genetically unique. And then anaphase, the same thing as meiosis. The main key concept of meiosis is that it just undergoes it twice. It comes out with 4 haploid cells, and it crosses over twice. So that's how we get genetically unique. Not all of these cells are the same. That's why for male sex cells, there could be the X chromosome in one and the Y chromosome in one. 2 of these ultimately will be X chromosome, and 2 of these will be Y chromosome for the male. And for the X chromosome will be, like the genes will be different on all of these chromosomes due to crossing over. So, yeah. cytokinesis, which is the actual pulling apart and separation of the cell. 4 genetically unique cells.